we had begun talking about, come to a topic where I said that, remember we're now at a 10,000 foot view, raw survey overview of the stuff that we've talked about like all semester long. And we're going to come back now pretty shortly, starting with the next chapter of lecture notes, beginning to get into the nitty-gritty detail. So we're still big picture zooming out. And I told you that we're going to be discussing, but I'm just telling you now as an overview, that lymphocytes, both T cells and B cells, they need not just one, but at least two, and I think most often actually three, maybe more, signals before they can actually get the full response to the antigen. They're going to then proliferate so that the one great cell that's got the right antibody on its surface, or T cell receptor, that's a precious cell. But the first thing we want to do is to have that cell start proliferating so that you form clonal descendants of that cell. Maybe we'll have 100 clonal descendants, and they've all got that sweet antibody or T cell receptor. Then having amplified the numbers of it, they'll then differentiate into armed effector T cells or so-called plasma cells that secrete antibody. To start and to do that whole program of proliferation and differentiation, you want to be darn sure that it's really foreign and not a self-antigen. You don't want to be launching an autoimmune response. Hence, at least two signals. So for B cells, the first signal they get is from when their surface antibody encounters a cognate antigen. That initiates a signal transduction event, which will start the process. But then a helper T cell, this will be a CD4 T cell, it may have already encountered its cognate antigen and then get into an actual full-grown helper T cell, or in certain circumstances, it can encounter it on the B cell, and then so a naive CD4 T cell can begin the process of proliferation and differentiation. But the main thing is eventually this CD4 T cell is going to have to be a full-grown T cell and is able to secrete the cytokines, small hormone-like peptides, for which there are receptors on the B cell surface. And those receptors, these cytokine receptors on the B cell, can initiate the second signal that you need to actually begin. Okay, all systems go. And similarly, a naive T cell here, it will become its T cell receptor. Well, again, I didn't elaborate on this over here, but the T cell receptors encounter their cognate antigen in the form of a small peptide as presented by a so-called MHC molecule. Again, MHC molecules here on the B cell are going to present peptides derived from the antigen protein to the B cell. Okay. B cells are one of the three kinds of so-called conventional antigen-presenting cells. So they can then activate T cells. The other second major antigen-presenting cell is the dendritic cell, for example, here. So this dendritic cell is encountered in the cognate antigen. It's very sticky on its surface, and it will bring foreign antigen in, cut it up into pieces, load some of the pieces, we hope, onto its MHC molecule, and present that foreign peptide to the T cell receptor on a T cell. That's signal number one. The T cell receptor generates signal number one. But then this cell, what makes the cell a professional antigen-presenting cell in a way is the fact that it expresses molecules with names like B17 or B7, I mean. This is a transmembrane protein that binds to a transmembrane receptor on the surface of the T cell, and that transmembrane receptor in the T cell then sends signal 2 into the nucleus. So then we moved on to say, so here we're going to do some DE stuff here. The MHC molecule, we talked We know antibodies are a big thing. We know that T cell receptors are a big thing. The third really big thing that we're going to be coming to again and again are the MHC molecules that present foreign peptides to the T cell receptor. So we introduced here some talk about the T cell receptor and how it encounters its peptide on an MHC molecule. So now a little bit more detail then. We said when it comes to the T cell receptor and the MHC molecules, we said that antibodies, initially in the form of a surface antibody on a B cell, which will be an IgM, M meaning it's a membrane protein. It can bind a few amino acids on the surface of a whole protein, okay, and then become activated. This IgM can then become a secreted antibody, recognizing the same epitope on the surface of that foreign protein. But T cell receptors, in blue, I said that they're evolutionarily related to antibodies. They represent sort of evolutionarily one of the antigen binding arms of an antibody molecule. But T cell receptors cannot just bind a free-floating protein, recognize a peptide sequence on the surface of a free-floating protein. It can't really bind to it. If it could, it would not be able to generate any kind of signal. Instead, it can only see the foreign peptide from this protein, let's say. This protein would be brought in, degraded into bits and pieces. Now, the protein is broken up into bits and pieces and loaded onto a yellow MHC molecule here. Is not necessarily, this peptide that's going to get loaded on, let's say, to this MHC molecule, that ain't necessarily the same peptide that has the antigenic sequence that the B cell recognizes. It could be any peptide derived from this protein, or from some other protein perhaps related to the infection or something like that. So this T cell, this one peptide out of many that would be produced, MHC molecules can't bind all peptides. They can bind many tens of thousands of peptides, but not the entire universe of all possible amino acid sequence peptides. And so we have to hope that your immune system, with your particular set of MHC proteins that each of you can make, is able to bind at least one peptide derived from a foreign antigen and present it on its surface to a T cell, and the T cell receptor binds to not just the peptide, but the peptide MHC protein complex. So, you kind of get the idea, but kind of not. It'll soon become clear. Now, last time, I showed you this picture, and I said we'll come back to it, and we're still going to have to wait to come back to it. Okay, so I'll come back to this again today, but last time, and again now this afternoon, I'm going to instead go ahead and hear a little bit more in more detail about MHC molecules. If you notice me that I've been standing for a long time, raise your hand and say, "Sit down, Dr. Burr." My wife will be glad because I'll be a human being when I get home. If I don't sit down, I'll be in pain and I'll be a snarling bear when I walk in the door. A wounded animal. Okay. I told you then last week that there are two kinds of MHC proteins that cells can make. Now, all cells make a class 1 protein. Only a subset of cells make, in addition to class 1, make a class 2 molecule. The cells that make this second kind of MHC molecule, these are these professional antigen presenting cells that I told you about just a minute ago. So these are the B cells. I said B cells, and presented by their MHC molecules. And I said dendritic cells can also, they're really par excellence, able to present peptides on their class 2 molecules. The third cell type is the macrophage. These are the professional antigen-presenting cells. And professional antigen cells, professional antigen-presenting cells, they make class 2 molecules and present peptides. They also, like everybody else, also make class 1 molecules. Now the reason, basically, that all cells make and express class 1 molecules is because the main job of class 1 molecules is to prevent foreign peptides derived from virus infections. And class 2 molecules, they mainly present peptides, well, they present peptides made from typically bacteria. Extracellular antigen is brought into these cells, degraded, and then put onto the MHC molecules. So this is antigen derived from stuff that's outside of the dendritic cell or macrophage or B cell, brought in and then loaded onto the peptide, brought back out and presented on the cell surface. This peptide is derived from cytoplasmic proteins, or maybe I would say cytosolic proteins. And it's virus infections we're talking about here. When the virus infects a cell, it starts to replicate and make new viral proteins in the cytoplasm of the cell. Those viral proteins, made off of the ribosomes in the cytoplasm of the infected cell, they're cut up into pieces and they are presented on class 1 molecules. So class 1 molecules essentially say to the immune system, as I said before, they say, come kill me. I'm infected. For the love of God and for society as a whole, stop me before I kill again. Okay? Okay. So here then, we had just, this is where we finished up now. Last time. Again, more about MHC molecules. This is a cartoon representation of what I've been telling you about. You can think of this as two different cells, or maybe just one cell. This one cell, it would then have to be a professional antigen-presenting cell, a macrophage or a dendritic cell, and it has become virus infected, but it's also bringing in, say, foreign bacteria here, and cutting them into bits and pieces. You could think of this as being an antigen-presenting cell. But it's maybe simpler just to imagine this could be any cell here. This is one cell here. And this over here is what would be happening only in a professional antigen-presenting cell. Everybody with me so far? Any questions so far? Okay. So, here we're looking at a virus-infected cell. The virus got in and was making viral proteins off of the viral messenger RNAs, and these viral proteins will then be used to form, one virus particle gets in, and it then produces what you need to make 100 new virus particles inside the cytoplasm, and then they're released to infect 100 new cells. Now, having said that, or I keep emphasizing that class 1 molecules their main job in life is to take peptides derived from intracellular virus proteins, load them up, put them on the cell surface, and say, "Come kill me." And killer T cells, CD8 cells that have differentiated become cytotoxic lymphocytes or killer T cells. They will be very happy to come kill this cell. They really love killing target cells. They're really ruthlessly efficient at doing that. But now let me just broaden this a little bit. I just want to say that at all times, all cells are grabbing normal cell proteins and cutting them up into bits and pieces and loading them onto class 1 molecules. So that at all time, all your cells are presenting peptides from any one of the tens of thousands of different kinds of proteins that they have in their cytoplasm. So that your normal cells, they've got maybe 10 to the fourth MHC1 molecules on their surface. And these MHC1 molecules are presenting self-peptides. And the immune system ignores them, as we'll be going into more detail, as I mentioned previously. The reason the killer T cells ignore these is because when these killer T cells were just being born in the thymus, they were exposed to all kinds of self-peptides on class 1 molecule like this. And any killer T cell who had made a shiny new gene encoding a shiny new T cell receptor is so happy and proud. I'm a T cell, I'm making a T cell receptor, but he recognizes the self protein, so he is killed in the crib, okay? Infanticide. And so he never survives. Only the CD4 T cells who've made shiny T cell receptors that don't recognize self are permitted to enter circulation. So your circulating CD4 T cells, they've been carefully selected not to recognize self-peptides presented by the class 1 proteins. They mean nothing to them. Okay. So, but if the cell becomes virus infected, then and now we're making viral proteins together with all the other 10,000 different kinds of normal cell proteins, and we'll start making viral peptides. We'll start cutting these viral proteins out. Okay. To my knowledge, there's no particular way that the cell can recognize a virally specified protein from a normal cell protein. They're all grist for the mill, so to speak. Okay. All proteins are going to be degraded and loaded on. So now, in the virus-infected cell, among the tens of thousands of MHC1 molecules presenting tens of thousands of self-peptides, now a percentage of the MHC1 molecules on the surface are now presenting viral proteins to the immune system. And they are recognized and killed, and the killer T cells then kill the cell. Kill. Okay. Now, I want to go into a little bit more detail. What happens to the proteins? That it's a puzzle for immunologists for a while. This is a transmembrane protein facing the outside world, and it has a peptide loaded onto it, which was derived from an intracellular protein. So how do intracellular proteins, when we break down intracellular proteins into bits and pieces, how do some of these bits and pieces get loaded onto a molecule that's facing the outside world? Well, so you might wonder, well, maybe you cut them into pieces and then they are transported to the outside world and then somehow diffuse around and they'll land on an MHC1 molecule. Well, that's not a very elegant idea. What happens is, of course, is these proteins, these viral proteins, or any cellular proteins, are picked more or less at random by a ubiquitination machinery. If you take in biochemistry, you learned about E1, E2, E3 ubiquitin ligases. Would you please raise your hand if these are not totally foreign words for you? You've encountered E1, E2, E3 ubiquitin ligase. Well, congratulations, good for you. I'll talk only briefly about that because I don't want to get into all kinds of details. Suffice it to say, for the purposes of our course here, that there's this machinery that will recognize certain proteins. Okay. The machinery involved here will bind to the foreign protein, and then it will grab, it will be presented with a small protein called ubiquitin. Ubiquitin is quite small. It's present in high concentrations in all cell types. Because it is ubiquitously expressed in all cell types, they decided to call it ubiquitin. For those of you who English is a second language, when I say something is ubiquitously expressed in all cell types, that means universally expressed. All cells express it. Okay. So this machinery will grab proteins, sometimes quite specific, often quite specifically targeted. You can start expressing certain components of the ubiquitin machinery that will specifically recognize certain proteins and cause them to be ubiquitinated, which is to say, that the protein will bind this ubiquitin machinery, will grab this red protein, hold on to it, and then grab a ubiquitin molecule presented by other members of the ubiquitin machinery, and take that activated ubiquitin and transfer it on to a lysine of the target protein. And then having attached one ubiquitin to the target protein, it'll hold on to that ubiquitin and then attach a second ubiquitin onto the first one. And then add a third ubiquitin onto the second one. So it'll assemble a chain of ubiquitins, okay? Maybe 10, maybe 20 ubiquitins all in a row, like a little whisker of ubiquitins that are growing off the surface of this protein. And then here we have a molecular machine called the proteasome. The proteasome, the walls of the proteasome here shown in purple, consist of a wide variety of different proteases with different specificities. And they assemble to form a tube-like structure, and there's a channel in the middle of the tube. And the foreign proteins, these proteins are the ubiquitin tag. On this end of the proteasome, we have machinery that will grab the chain of ubiquitins as this protein goes drifting by, and the chain of ubiquitins brushes against the open end of this barrel-shaped object. Proteins here will grab the ubiquitin and then wheel it in, and then unfold the red protein to a linear amino acid sequence and stuff that linearized protein through the little channel in the middle of this, where it is then sliced and diced as it's moved through the proteasome. So whole unfolded proteins come in this end, and bits and pieces sliced and diced of the protein emerge out this end. Peptides that have just the right size, that are in the range of the right sizes to be loaded on the MHC molecules, are then preferentially recognized and transported across the membrane of the rough endoplasmic reticulum. So this is the rough endoplasmic reticulum here. And newly made proteins are in here. Now, I guess it doesn't maybe have to be the rough ER, but it's rough, smooth ER regions here. And here we have a newly made molecule of MHC, an MHC1 molecule, shown here in yellow. It's just been read off of the ribosomes, membrane-bound ribosomes here, and then by co-translational translocation. How many of you have had biology 3302, eukaryotic molecular cell biology? Raise your hand if you have taken 3302. Raise it high so I can get a better sense. Not as many as I thought. All right. Well, even in introductory biology, you may remember, I'm sure you were told, that certain mRNAs coming out of the nucleus, our ribosomes will jump onto them. And the ribosomes will start running out the messenger RNA, reading it into a sequence of amino acids. The sequence of amino acids that's being made, it comes out amino terminal N first on the bottom of the ribosome. If this is a protein that we're making here that's supposed to become a membrane protein or a secreted protein, as the newly made polypeptide chain emerges from the ribosome, there's a little sequence here, like this little eraser end here, that's recognized. It's grabbed by a recognition particle. The whole ribosome mRNA complex will then diffuse around and be docked receptors in the membrane of the rough ER. And then we resume translating the mRNA, and the newly made protein is extruded through the translocon pore in the ER membrane. And now, so it comes into the lumen of the ER, where it will finish reading the whole mRNA, and the whole protein is now extruded into the lumen of the ER, where there are chaperones that will help it to fold. Okay. Now, I'm giving you some detail here. This would be a whole lecture in Bio 3302, and I'm giving it to you in about 2 minutes here. I'm not going to expect you to reproduce this story that I just told you in this class. I want to give you some sense of the story here. So here are newly made MHC molecules here. Now these newly made MHC molecules any protein that's made by this process here of co-translational translocation, okay, across the membrane of the it must stably fold to its finished shape before it can then be released to the Golgi compartments, okay, and then be either extruded, exocytosed, or if it's a membrane protein, now become a plasma membrane protein on the cell surface. These newly made proteins have to be fully folded. MHC1 molecules are still loosely folded until they get a peptide loaded onto them. But of the many peptides that are being brought across here, some but not all of these peptides will be able to bind to some but not all of the MHC1 proteins that each of you makes. I'm going to get into this detail later on, it'll become clearer. But here's an MHC1 molecule, lucky guy. He's able to bind this particular little peptide here that's being produced by cutting up these proteins here. It can now stably fold, it's the peptide MHC1 complex. It is stably folded, and it will then go by vesicle trafficking through the Golgi pathway. And here's this vesicle containing the newly made MHC, facing the interior of it. Here's the peptide. And the membrane of this vesicle fuses with the plasma membrane. This, which was facing inside the vesicle, is now facing the outside world. If you're confused about topology here, it's very simple. I'm not going to trouble to stop and explain it to you now, but you can pick up any cell biology textbook and there are pictures that make it very clear. If this membrane here fuses with this membrane, and like this, you can see that all this is now, any fluid, any soluble components are going to be released to the outside world. This membrane protein is now a membrane protein. It was in the lipid vesicle membrane. It's now in the plasma membrane, and it's facing the outside world. So basically, the peptide that's going to be loaded on, it gets brought across all this in white, essentially topologically outside world. All that's gray here is topologically inside world, so to speak. So this is where the peptide gets transported across a membrane from the inner world to outer world, gets loaded onto an NHC molecule with this whose binding component here is in the topologically outside world movement of the ER, goes by vesicle trafficking, now it's facing the outside world. Okay. Now, I've been at pains to kind of go through the details of this. As I just said, I'm not going to ask you all the details of this, but you're going to remember that peptides derived from virus proteins get loaded onto MHC1 molecules. They're first going to be transported by a TAP transport machinery. Okay? They're going to get peptide loaded onto them here, and then go by vesicle trafficking, and now appear presenting this peptide, which is derived from intracellular protein, to the immune system in the outside world. Questions? Okay. Now, however, if this were a professional antigen presenting cell, then we're going to have this kind of pathway. Now, again, a professional antigen presenting cell, like a dendritic cell, it could be virus infected. And it, like all cells, it would be you doing this pathway to present viral peptides on the surface. But professional antigen presenting cells have the extra ability of being able to phagocytose extracellular antigen. Could be just a foreign protein, or a whole bacterium or a whole virus particle is brought into the cell. Okay. Lysis is what I've drawn here is a little vesicle here, and this is not a check mark, but a capital L, where you kind of have an angle here. Capital L for lysosome. Okay. So this lysosome here has digestive enzymes, and that's what lysosomes are, the new molecular stomachs that all cells have. This has it's an acid pH, and it has digestive enzymes. This lysosome membrane will fuse with the endocytic vesicle or the phagosome here. Okay? And now all these digestive enzymes are able to work on the bacteria or virus particle that's brought in, or protein that's brought in. These membranes here also have proton pumps, and they're moving protons from the cytosol into this compartment. So this compartment is acidifying on its own, but also the lysosome was already a very acid material. When these two compartments fuse, you'll get acidification just by the fusion of the acidic pH interior of the lysosome. So now this is an acidified phagosome here, and we're digesting this foreign particle into bits and pieces, by which I mean into peptides. And these peptides are going to be loaded onto a class 2 molecule. Those class 2 molecules have 2 transmembrane domains here. So this is a class 2 molecule. Now this class 2 molecule was made just by co-translational translocation, just the way class 1 molecules are made. So here's a newly made class 2 protein. Okay. Why doesn't it find these peptides that are coming into this compartment here? It doesn't. How come it doesn't? Well, that's because there's a little protein here called I sub I, which is an abbreviation for invariant chain. Do not ask me why I sub I is the abbreviation for the phrase invariant chain. Why would it be chain sub I or something like that? No, it's I, big I, sub little I. And I sub I, or invariant chain protein, is shaped kind of like a walking stick or a candy cane, and the handle of the walking stick, so to speak, sits right here in the groove, on the peptide binding groove of this MHC molecule here. So the peptide binding groove is already occupied by this II chain. So these little guys coming from intracellular proteins, they're coming into this compartment. They can get onto class 1 molecules, but they're blocked from getting on to a class 2 molecule by the invariant chain protein shown here in red. And the invariant chain, by binding to the groove, stabilizes the folded conformation of class 2 molecules. So they are permitted now to leave the ER compartments and go by vesicle trafficking through the Golgi compartment. Now, most stuff coming through the Golgi compartment fuses directly with these vesicles coming off of that. Well, that the default pathway normally is to fuse to plasma membrane. But this vesicle has certain other proteins on its surface that prevent it from docking with the inner surface of the plasma membrane. It instead wanders around and then can find that it can, this membrane can bind and fuse to the membrane of this thing here. So this guy is fused with this, forming, here's an fusion here. When these two vesicles fuse, here's the class 2 molecule now in the lumen here of this phagosome, and all these peptides derive from extracellular antigen, class 2 molecules bind extracellular antigen. They get loaded on, and now, sort of hocus pocus here, the class 2 molecule with its peptide derived from extracellular antigen will now come up here. This membrane will fuse with the plasma membrane, and now this protein is now facing the outside world. And presenting class 2, it's being presented by a class 2 molecule here. And while CD8 expressing proteins can bind class 1 MHC peptide complexes, CD8 proteins cannot bind CD4 class 2 molecules. The CD4 T cells, their T cell receptor can bind a class 2 molecule with its foreign peptide. And so MHC2 molecules activate CD4 T cells to become T helper cells. Class 1 molecules can activate CD8 T cells to become killer T cells, or if they've already become killer T cells, this T cell receptor in a CD8 cell can bind only a class 1 molecule. And we're going to talk about why that is in a subsequent lecture. But it's only the T cell receptors in CD4 T cells that can bind productively this MHC2 molecule with extracellular, typically bacterial peptides, and class and CD8 T cells, killer T cells, they're only able to bind peptides on class 1 molecules. So should maybe, we're going to go back to this in more detail. Next Friday, I'll finish up the material that we're going to be talking about here. And then I'm going to take 15, 20 minutes, maybe more, and kind of go through the lecture 1 notes here and some of the class 2 notes here. And I'm going to say, don't worry about this page. I'm going to say, on this side of this page 3 here, be sure you know this. On page 4, don't sweat it. Page 5, don't sweat it. Page 6, be sure you know this. Okay, so I'm going to hold your hand. I'm going to walk you through, okay, what you need to know for the test. Okay, I'm a kind person. So, but I know many of you are already beginning to kind of freak a little bit because we've covered a whole lot of stuff here in this broad overview. So, as I keep saying, not all of it, all that detail is going to be on the test, but sort of general ideas and type stuff. And there'll be, when we start getting into more detail here shortly with the class 2 lecture notes, now I'm going to ask you about some of the detail that's on this. And I'll be telling you as we go through this what you need. But just broadly speaking, for the first exam, you should remember MHC2 proteins present peptides derived from extracellular antigen. I could not believe that. Flashing yellow lights here. And class 1 molecules present peptides derived from intracellular proteins, typically virus proteins. Any questions so far? Now that okay, this looks complicated, but it's not basically very complicated. I want to give you the main idea here. Here in red, we have these red guys here represent genes. This is a DNA molecule. It's like in humans, this would be chromosome number 6. Okay. And this is actually what's called the histocompatibility locus here. In this one chromosome, there's one region of this one chromosome. We've got a whole bunch of genes involved with the processes that I've been telling you about. Human beings have 3 genes, conveniently called A, B, and C, that code for class 1 protein molecules. And conveniently, we have 3 genes that code for the proteins you need to make class 2 molecules. Class 1 molecules, these genes here, remember the class 1 molecule has one transmembrane segment and kind of a supporting domain here, and then a little platform domain here where the peptide is going to rest. That's what these 3 genes code for, this major component of a class 1 protein. There's a little smaller component that kind of sits interacting with the base stuff here and propping up the platform. This is called walking on the word for the moment. Don't worry about that. Beta-2 microglobulin. This little guy here is beta-2 microglobulin. And it's a weird little protein. This is its only function as sort of a secondary cast role supporting the main guy here, the A, B, and C protein here. Class 2 molecules, you may remember, they have, perhaps I should go back and refresh your memory here. This is the class 1 molecule. One large protein here has the base and the peptide and the platform of the peptide and beta-2 talked about is just kind of support role. Class 2 molecules, we have two alpha-like proteins here. Here's alpha itself, here's beta, and together they provide the sort of supportive structure here, and then together they make the platform that the peptide is going to bind to. So it really takes an A, B, or C gene only to make what business end of a class 1 molecule. This could be an A gene protein, or a B gene protein, or a C gene protein. Here we have two genes per class 2 protein, okay? And so here, the three genes that code for class 2 proteins are called DP, DQ, and DR. Everything, the broad ideas come from just talking about class 1. It's a little bit weird to start getting into the class 2. You have, for the DP class 2 molecule, you need an alpha chain and a beta chain. Okay? And for DR, an alpha chain and a beta chain. And for DQ, and then for DR, you have an alpha chain, and then you can choose between either of two beta genes here. So this kind of begins to muddy the water, but don't worry about this. The basic idea is that we have three genes that make class 2 molecules, and we have three genes that make class 1 molecules. Okay. And again, we're going to turn to this. These genes, we have evolved, vertebrate animals have evolved machinery that makes, when these make sperm and egg, okay, we have a higher mutation rate for these A, B, and C genes. So that in populations, we have, these genes are, we say, genetically polymorphic. Okay. So my A gene that I got from my, so we're diploid organisms. I got this chromosome from my mother, and I got the corresponding chromosome from my father. My father's A genes, slightly different than probably an amino acid sequence, from my mother's A genes. So even though there are only 3 A genes per chromosome, because I've got 2 copies of every chromosome, I have actually 6 genes that code for class 1 molecules. I've got A from mother and A from father, B from mother, B from father, C from mother, C from father. So I've got 2 each, A, B, and C. And probably my mother's A is genetically a little bit different from my father's A. That's just because of this process that evolved over evolutionary time to make it so that my mother and father's A genes are probably different from each other. And my mother's A gene can bind one set of peptides. My father's A gene can bind a different set of peptides. In the entire universe of peptides, each MHC protein can bind maybe 10,000 out of the universe of all peptides. So my A gene can bind 10,000 peptides. My mother's A gene can do that. My father's A gene can also bind maybe a completely different set of peptides, maybe an overlapping set, but very likely significant differences. So maybe I can find a total of 20,000 peptides out of the universe of countless millions of possible peptides by my A genes. Same thing with my B and same thing with my C. Okay. That's good. Okay. The same thing was true for the Native Americans when the Spanish conquistadors arrived and so forth. But none of their, in their population, very few people had an A gene allele that was able to bind a smallpox peptide. Okay? They could bind a million other peptides from a million other pathogens endemic in North and South America, but they hadn't been selected for MHC genes that could bind, make proteins that bind a smallpox peptide. So 99% of the Indians got wiped out, and the few that survived, then their children survived. So now the Native American populations in the Americas now, they've been selected for the ability to produce variant A or B or C genes that can bind smallpox and measles and so forth. Okay? The Europeans went through this process 10,000 years ago when they first, you remember the story, they were able to domesticate animals and plants. So the earliest cities and towns of the globe were in the Middle East. The animals they domesticated had animal versions of measles and smallpox and so forth. So the Middle Eastern populations suffered the same fate as Native Americans. It just happened to them 10,000 years earlier. They got wiped out, and the survivors were those rare people who perhaps would be able to have a mutant gene that could produce A, B, and C. And the reason we all have this huge genetic polymorphism of these genes is precisely because we want at least somebody to be able to present a Martian peptide from a Martian virus that arrives on Earth at some point down the road, okay? So that we don't all suffer the same fate as the Native Americans when the European Martians arrived and with their European viruses. So it's good diversity, such a commercial for cultural diversity is good, right? We all know. Okay, so you get all that. So we have 3 class 2, P, U, D, R. We have 3 class 1, A, B, and C. Okay. But we have mother and father, so we can typically make 6 different class 1, 6 different class 2, class 3. Another name I told you before. I'm using the generic name Major Histocompatibility Complex, MHC1. Okay. So these genes and the proteins are called the MHC genes, MHC1 genes, MHC2 genes. So we speak of the MHC1A gene or the MHC1B gene. In medical terminology and sometimes in some immunological laboratories for historical reasons, these genes and the proteins were called human leukocyte antigens. So often in a hospital environment, you'll hear these referred to as HLA genes and HLA antigens. HLA. I think I said somewhere that HLA stands for human leukocyte antigen. So HLA-1A is the same as MHC1 gene. And these genes, as these ones too, are very polymorphic. This is genetic polymorphism. The nucleotide sequences of this, of the A gene from my father, are probably different from the nucleotide sequences of the A gene of my mother, and therefore the proteins that are encoded are a little bit different. And the amino acid differences are always in the little part of the protein that binds the foreign peptide, not the base and all of this. Always concentrated in the part that binds and presents the peptide. And therefore they govern the specificity of what subset of the universe of peptides can be. Are you still having fun? Okay. Keep on going here. This just summarizes what we've already said, that class 1 MHC molecules, they bind peptides derived from viral infections and virally specified proteins now in the cell. They get chopped, sliced, loaded, and transported and loaded onto class 1 molecules that then go by chemical trafficking and present these peptides that came from intracellular proteins from virus infections. Okay? MHC1 proteins are all about dealing with virus infections. And then killer T cells, they can recognize these class 1, these forms of class 1. Now, this already gets us right to talk about more. The reason that killer T cells can recognize foreign proteins on class 1, but not on class 2, is because these killer T cells express the CD8 protein. When they first come out of the body, we call them CD8 T cells because they express the CD8 protein together with the T cell receptor. And the CD8 protein acts as a kind of a co-receptor here. The T cell receptor binds the foreign peptide and the MHC stuff in the vicinity of the peptide. The CD8 protein is soluble in the membrane, never too far away from the T cell receptor, and it can snuggle in here and it can find the base of the MHC1 molecule here. Class 2 molecules do that. CD4 proteins can only, class 2 molecules can't be recognized by CD8. Class 2 molecules are recognized by CD4, and the CD4 protein plays the same role in CD4 T cells. I'm getting confusing here. I'm getting ahead of myself too. So here's the deal. Class 1 molecules are presenting viral peptides derived from cytoplasmic viral proteins, and then they're recognized by CD8 T cells that become killer T cells, and they then send down hellfire missiles on the surface. So, yes?
So do CD4 T cells recognize MHC1?
Not at all.
Not at all.
In fact, there's a picture I'll show you. This comes right next. So here's the deal. Again, summarizing what I've already been telling you. Here's a professional antigen presenting cell. In this case, it might be, for example, a macrophage. And the macrophage engulfs whole bacteria. It's brought into the cell, the phagosome here. The lysosome fuses with this, and the lysosomal enzyme then slice and dice this bacterial cell into peptides, okay? And then a vesicle that came from the Golgi compartment with newly made class 2 fuses with this, and these extracellular antigen proteins are loaded onto the class 2 molecule here, and they're now presented on the cell surface. And then CD4 cells, as I said, the CD4 protein plays a role analogous to the CD8 protein, okay, in the sense that it recognizes the base of an MHC molecule, whereas the CD8 protein recognizes the base of a class 1 presenter. The CD4 protein recognizes the base only of a class 2 MHC molecule. So CD4 cells can only interact with MHC2 proteins, and the CD8 T cells can only interact with class 1 expressing cells. Okay. So a T helper 1 cell here recognizes this foreign protein here, okay? And it supercharges these phagosomes here. Some bacteria, when they find themselves in this environment, they say, ha ha ha, bring it on, oh, make it hotter, I love it, okay? They're quite content that they just grow happily inside of here. Well, in the arms race, the response to that has been to have T helper 1 cells, when they encounter this, they're going to secrete cytokines that turn on the expression of like industrial strength bleach expression, okay? So we really bleached the Jesus out of these bacteria that were living happily, eating hot chocolate, field grapes and so forth, and loving it. Now suddenly things get really much too hot for them, and they're killed inside of their previously happy little phagosome environment. So that's the role of helper 1 cells, to get rid of bacteria that love to live inside of cells, like tuberculosis bacteria, leprosy bacteria. They get into macrophages and they love to live in the phagosome here until helper 1 cells come along and activate a system that will make it impossible for them to be happy. Helper 2 cells, again, both of these express the class 2 molecules here. But because the cytokine environment is informing these cells, they have become helper 1 or helper 2. The helper 2 cells, remember we said, their CD4 protein permits them to interact with T cell receptor to interact with class 2 molecules presenting bacterial peptides. Bacterial peptides derived from, for example, the foreign protein that the antibody had bound. The antibody binds this foreign protein. The whole antibody antigen, the whole antigen antibody complex is brought in, all digested into bits, and the peptides derived from the foreign protein here are then loaded onto these newly made class 2 molecules. Okay. And now these T-helper 2 cells, when they see these peptides derived from extracellular foreign protein, they then give the second signal that the B cell needs to become a plasma cell that will then secrete this antibody. So what good are antibodies? Well, antibodies are helpful. Antibodies, some of the antibodies that you make are specific to viral surface bacteria or viral surface proteins. So they coat the surface of the viral particle or the bacterium, thereby opsonizing the bacterium. Okay? And then macrophages can the oxidized bacterium is much 10 times more efficiently engulfed and degraded by macrophages. Macrophages, antibody bound to antigen here also activates the complement thing here, and we start depositing complement on the surface of the bacterium, and we have complement receptors, so complement fragments also oxidize whole bacterial, whole large particles here. Also, some bacteria, they have to bind to the surface of the cell when they get into the cell, into bodies and so forth. The antibodies that coat the surface of the bacterium prevent it from binding receptors and getting into cells. Also, many pathogenic bacteria secrete proteins which are toxic, and antibodies will have been made against the the bacterial toxins here. Here the antibodies binding to toxin molecules here, and the antibodies bind the toxin just right. The toxin's not able to bind to the normal receptor like you get inside the cell. So antibodies can neutralize bacterial toxins. Now then, also, so all of this stuff, MHC1 T cell receptors, B cells and T cells, this is all the adaptive immune response. Okay? These T cells that make T cell receptors, they're the 1 out of a million T cells will just the right T cell receptor to fight the infection. Those T cells proliferate and fight the infection. Most of those T cells then go away quietly and kill themselves, but some of them stay on as memory T cells. Okay? Same thing with B cells. So that if you're successful, you get infected, there's a several week lag phase here. Come on, innate immunity. We're depending on you innate immunity by the time until finally the T cells and B cells start coming into play, and they can then defeat the infection most of the times. And then these B T cells die off, but some of them stay as memory cells, and then if you encounter that same pathogen, boom, very rapid response, very strong and rapid response. So oftentimes, if you were to encounter that same pathogen, get a little infection, get it started, you never even feel the symptoms. You just cure it up before you even die. So this is long-lived immunological memory. And of course, this is the basis of vaccines. Okay? We vaccinate the child not with the whole nasty bacteria, but with little bits and pieces of the bacteria, or killed bacteria, and then we make on-receptor T and B cells. They fight off little bits and pieces. It's not really an infection. But now we have memory cells against the bits and pieces, so that if you get the real bacteria or virus in, they're expressing those bits and pieces as part of their normal proteins and so forth, and the memory-making T cells spring to life and you're protected from infection. And I'll just say a few words here, and we'll take our break. We could probably take our break right now because nothing on this page is going to be on the first exam. But let's just go through it. This is, so all that we're talking about so far is how the immune system protects us, and it really does. It's a fantastic thing. But things can go haywire, and so if you get either hereditary or whatever problems with your immune system, then I think it might feel like miserable and even short-lived. Okay? So the immune system can be compromised by inherited immune deficiencies. Okay? The famous bubble boy in Houston, this is after you, this is before your time. There was a Seinfeld episode about the bubble boy. Did anybody see that Seinfeld episode? It turns out this child is a real thing. He was recognized as having this disease, and they got him into a sterile environment. And then they moved him into an actual sort of spherical, oxygen-filled, air-filled plastic bubble with ways of bringing food in and out and so forth. And he lived in this thing for a number of years, all the while they're hoping to have some ways of treating the disease. But eventually the bubble was breached, and he actually, maybe I think they tried experimental treatment on him, and he died. In the Seinfeld episode, the bubble boy is an obnoxious little boy. And George was kind of an obnoxious little boy himself, all grown up. He and the bubble boy get in a fight together. George breaches the plastic bubble. Anyway, Seinfeld humor. So we have inherited diseases. There are also inbred experimental mice. These are mice that have a mutation in a gene that's needed for the so-called WIN gene. This gene is needed to make a functioning thymus. Okay, it turns out it's also needed to make functional hair follicles. So these mice, they can't make a thymus gland, and they're also nude mice because they can't make any hair. Because they can't make a thymus, they can't make T cells, and they accept transplants and grafts very easily. They're widely used in cancer research to transplant them with certain tumor cells to see how the tumor is going to grow in vivo environment kind of thing. But you can also get certain pathogens or toxins that can reduce the immune system. And the famous case here, of course, is the HIV virus. Okay? The HIV virus specifically targets the CD4 protein on CD4 T cells, and CD4 T cells are able to be infected by the HIV virus selectively. The HIV virus, when it infects a CD4 T cell, kills it. First it replicates and then it kills it. Your immune system is not capable of defending itself against the HIV virus. It's a very insidious virus. We're not able to get vaccines against it yet successfully. If you get infected, after anywhere from 5 to 10 or 15 years, most people 5 to 10 years, your body can no longer replace the CD4 T cells that are constantly being infected and killed, and so your CD4 T cell counts begin to go down. And when you have these CD4 T cells, you can't protect yourself against all kinds of opportunistic infections. And so you die miserably from all kinds of infections that you really, without an immune system, even antibiotics and so forth, if it's bacteria, if it's viruses, you're really screwed. Antivirals. So you die because your CD4 T cells fail. You then die of infections of the virus, killing you. So that's problems with the immune system. On the other hand, you can also have a misdirection of the immune system, okay? So when misdirected against normal, harmless materials, okay, in the human body. The immune system can cause allergies, or even worse, autoimmune disease. And some examples of body failure because of an autoimmune, the body turning against itself. There's a lupus is a genetically, well, serious talk. Anyway, in lupus, your antibodies start making antibodies against your own DNA. Your own proteins. That ultimately fatal usually. Type 1 diabetes is the loss of the pancreatic beta cells that make insulin. They're lost because you have these individuals form an autoimmune response against the pancreatic beta cells and kill them. So eventually, over time, you have no more cells that can make insulin. And rheumatoid arthritis is something that sets in for people in middle and older age. Okay, it's terrible. It's a wicked form of arthritis. And then multiple sclerosis is an autoimmune reaction against certain proteins that form the insulation around your neurons, so your neurons start short-circuiting and so forth. Now then, for an increasing number of diseases, the replacement of tissues or organs by transplantation can now be done. The major limiting transplantation, the major factor in transplantation, has been tissue incompatibility caused by the extensive polymorphism of these class 1 and class 2 MHC proteins. Remember I said my mother's A and my father's A proteins are going to be different from each other? All that polymorphism is good in the sense that if you get a virus from Mars, at least some of us will survive. But it means there's a lot of tissue differences. I can't just get a liver from anybody. My best chance is to get a liver from one of my brothers or sisters, basically. Okay. So, but even there are going to be MHC differences. And we can now overcome those differences. And if you get a pretty good match, you can now use these new wonderful drugs. These are our generation's equivalent of penicillin back in the 1940s. These are drugs that have names like cyclosporin and tacrolimus and rapamycin and so forth that suppress the immune system to the point that they will not reject pretty well matched, but not perfectly matched donor organ. Of course, when you're taking these drugs, even though you're not rejecting the new heart or the new lung, you're at risk for other infections now because you're suppressing the immune system. Okay, so that's the end, except we never did talk about this page. And let's take a break though. This page kind of puts it all together, what the stuff that we've been talking about. And I do want to talk about this a little bit before we move into delving more deeply into these cells and anatomy.